What am I doing today? That is the question. So, what am I doing today? That is the question. Hi, Alex. Sorry, I was running a bit late tonight. Super excited. I love that. I love the fact that you're super excited. Right, hold on one second. I'm going to have to do something that I haven't done. Right, hold on. Okay, go. Hi, guys. Welcome. Give me 1 second. I'm just setting up for tonight. What am I doing? That's what I'm looking for. Right. Okay, whilst I set this up quickly. Nice. That's really cool, Alex. I'm really pleased for you. Right. There we go. I will do that. There we go. I'm hoping that will all have worked. Hi, welcome. Sorry, guys, I'm just setting up on the other screen. So everything module 2 and 3, you are. Hi, Andreas. How are we tonight? Right, I'm going to see if this works. It's annoying. Last time I did the pop out for the YouTube, it didn't actually show me any of the messages that people were writing there, so I completely missed it. You have your A-level theory paper on the 15th of May. So you're doing your AS paper. Okay. Waking up early, why? Because you're back at school now. You had some time off, so it meant that you were all in the whole relaxing late mornings. Yeah, I get that feeling. Although I have two very young children who wake me up constantly very early. So it means that I don't get that luxury of lying down and staying in bed. I wish I did. Okay, so I so far have a few mainly AS students here tonight. Do you like it? I actually did that myself. I was sitting with the girls. Thank you, Jinema. I was sitting with my girls and they were wanting to do drawing, and we've got these coloring pencils, which are watercolor pencils. So I was like, oh, I wonder if I can draw something that actually would work for making a video. And that's what I did. So yeah, that's my picture that I drew, and I used the watercolor pencils to make. So I love that. Okay, so do we have specific questions that you would like me to cover, or would you like me to just go through different questions? So I have a few of the ones we were looking at last session that we could continue on with. So there's one specifically that I was going to do. Hello, I was wondering if you could explain what we need to know about gene sequencing. Yes, Kate, I can go through that one as well. Kate, if you want to, I am on my YouTube live as well, and I've got actual screens there, and you can join the Zoom lesson if you would like. I want to cover this question first because I do want to get a video out there on this question. So I've been doing this question a lot with some of my private clients. And I'll answer that question in a second. So it's a really nice question, and I want to help as many people as I can with how to answer it because I've had a lot on serial dilution, and even I've been struggling with it. I've been back and forth and trying to get my head around because the maths I really struggle with. So is it true that biology at uni says everything at A level is wrong? It's not that it says it's wrong, it's that basically we have to try and teach it to you in a level that is accessible, and then they go into more detail because you can't take all the information. So it's kind of like we build the foundation for the next level to build more of that. So they will tell you bits that are not quite right, and then other bits moving forward. Would you benefit from for A-level biology at XLB? At XLB, there is a lot of overlap of the content, Zed, but the one thing I would say is your questions are much harder. You have less content to learn, but much harder questions because they're more application based. I do tutor a student on EdXLB, so I am constantly going over that topic area with them. And it is one of those things that I would say your questions are harder. Okay, so let's start this question then, because I would like to cover what does it mean. I feel like batch and continuous fermentation is potentially going to come up. So one of the things that's really interesting is there is an expectation that you know for the industrial scaling of microorganisms that you can do either batch fermentation or continuous fermentation. So I'm going to come back to this in a minute, but what I want to do is I just want to remind you at the bottom of the screen, we've got some additional space. I want to remind you of the fact that what you can do is think about it in a sense of if I have my population growth curve and I'm growing my population, what I need to remember is the different parts to it. So I have my log. No, I don't. I have my lag phase. Then I have my log phase. Then I have my stationary phase. Then I have my death phase. So I have my lag phase in my population growth curve because the microorganisms need to get used to the environment, get used to the substrates that they're going to be using. And so because you have that they're getting used to the environment, it takes time for them to multiply. So if we were talking about prokaryotic organisms and we're talking about them going through binary fission, we know that they are going to replicate, and that could take 20 minutes for each replication, but we have to remember that they are getting used to the environment first. Our log phase is our exponential growth. So this is our exponential growth. And the reason it's exponential growth is because they have all the right conditions that they require for the active processes in their life. Now, this is important for you to note that during exponential growth, they are going to be making their primary metabolites. Now, because they're making their primary metabolites, they are the different substances which are needed for the active growth phase. So this could be, for example, that they are needing to produce different proteins. This could be that they are needing to produce proteins that allow them to continue to grow and do everything that is necessary for their survival. So we're talking about this exponential growth. We're talking about them doing the metabolic processes and that metabolic processes that are necessary for active growth. Now, I'm going to jump to some year 12 content here and remind you of your cell cycle. So if we were looking at your cell cycle and we were talking about G1, S, and G2, we're therefore bringing in our growth phase where we have protein synthesis occurring. So the protein synthesis that is occurring during G1 is for things like proteins that are required for the cell to function. So when we talk about primary metabolites, we're talking about maybe some proteins that are necessary for the active growth phase. Now, when we get up to the stationary phase, what we're saying is at that stationary phase, we have got the fact that the level of the nutrients that are available, we've got the level of the, sorry guys, one second, I'm just moving. things across. Here, I can see some chat now. I couldn't see it a minute ago. So yeah, so we have our primary metabolites. We get to the stationary phase. When we get to the stationary phase, what are we doing is we're seeing that our resources are decreasing. The organisms are producing waste products. And those waste products are going to be within the environment. And what's going to happen is there's going to be competition for resources. So during the stationary phase, what they tend to do is they tend to produce their secondary metabolites. So we have our secondary metabolites being produced during the stationary phase. And when we get to the death phase, that's where there is too much buildup of our toxins. There are limiting factors which are affecting the growth. So therefore, the death rate is greater than the reproduction rate, and that is resulting in the numbers declining in the population. So it's important we note our population growth curve. It's also important that we're aware of when the primary metabolites are happening and when the secondary metabolites are happening. So if we go back up then and look at our question here, it says on industrial stage, try that again, in industrial scale, our microorganisms can either be batch fermentation or continuous fermentation. And they want to know, do you know the difference between the two? Well, the first thing I would do is add some notes in to this. Now, what topic am I doing? I'm looking at module 6 and I'm looking at the biotechnology module. So the first thing I would do is the continuous fermentation is continuous because we are continuously adding nutrients. What we're also doing is we are continuously removing waste products, as well as removing desired substances that we want, as well as removing any dead cells. But we'll also be removing some of the live cells. So we're going to actually have to also input new colonies to keep that going. So when we do continuous fermentation, we're constantly adding, we're also constantly removing things. So if you use that then to try and answer this question, waste product is removed during the fermentation process. Well, the waste product doesn't get removed from the batch fermentation. In the batch fermentation, we literally grow it until we have it at the point in which we want it, and then we are going to then filter it and process it later on. So therefore, this would be our continuous fermentation when we look at that question. So then what we want to do is we also want to look at the next one, which is a fixed volume of nutrient medium being used. So where would there be a fixed volume of the nutrient medium? So 1 second, I am just going to switch over and double check. There we go. Do I have access to the 2024 papers? Yes, I do, but I will not share them because they're not allowed to be shared. What is the best way to revise for this? The best way to revise for it is basically to look at the stuff that you hate and go over that and spend the time on that. Even if it takes you longer than you anticipate, it's important to spend time on it, because what you're doing by spending the time on the stuff that you hate is you're actually revising the topic area you need to succeed. So it is important to do the stuff that you hate first. Okay, so moving back to this question then, what have we got? Lyra, the key thing about application is ensuring that what you're doing is you're trying to constantly find where your content fits in with the question that's in front of you. So if I go back to this one, we have a fixed volume. So if it's a fixed volume, it's going to be the batch fermentation. When we look at the next one, the secondary metabolites are more likely to be produced. Now, remember, secondary metabolites are going to be produced during the stationary phase. Well, if we're continuously adding in new nutrients and we're continuously removing waste products, then during continuous fermentation, that should stay in the exponential growth phase rather than in the stationary phase where those secondary metabolites are produced. So in that case, that's therefore going to mean what we want to look at is the secondary metabolites are more likely to be produced during that batch fermentation, which also leads us to the second one. Where's the growth rate tending to be faster? Well, the growth rate would be faster with the continuous fermentation, because if we're continuously giving the nutrients, then what we can then do is we can then be able to keep that at the exponential growth. Sorry, Sean, you're asking a question about the 2024, you're stuck on one, which makes me assume that you're a year 14 student. I can't display it because that's obviously not fair, because there's going to be teachers who use it as mocks, etc. But if you ask me the question that you are stuck on, and send me a email on it, I'll have a look at why you're stuck on it, but I wouldn't want to do it on here because that goes against what the exam board wants us to do. They're under lockdown for a reason, because as a teacher, if I want to assess where my students are currently right now so that I can make predictions for their UCAS grades, I'm not going to want it to be that they are disadvantaged because someone has leaked the papers. So the reason the exam boards keep them under lockdown is to allow teachers to have something to be able to use to gauge where their students are actually at. Don't tell them what came up, although you can look at lots of videos that people put out there anyway, but it is about the understanding how to answer the question. Is it possible to go from a D to B by the exams? Yes, it is, as long as you're doing the revision the right way and you're finding the gaps in your knowledge and you're then using the time to fill those gaps and not just revising the same content again. Ooh, what's a flash goal? I have no idea what that is. Okay, back to this question then. The culture is grown for a fixed period of time. That is a fixed period of time. It's obviously your batch. So I would suggest if you are looking for things to revise, I would definitely say microorganisms, batch fermentation, and continuous fermentation is potentially going to come up, would be something that I would predict. But it's this question that I want to cover with you guys today, because I really want to do a video on it. I haven't had time because I've been busy tutoring all day. I've had a full day of teaching, so therefore it means I don't have time to do some of the other stuff I would want to do. So this question, though, I really like it because it makes me stop and think about how I can help students to understand the zero dilution question. So zero dilutions always come up. They come up pretty much every year, at least one way or one form or another. So obviously we were talking about the batch fermentation and the continuous fermentation. Well, for us to be able to grow our microorganisms, we're going to have to culture them. And so when we culture them, we need a way of figuring out potentially how much we have and how big that population is growing. The other thing we can do, which I do have a feeling is going to link together, is we could genetically engineer our, for example, microorganisms are bacterial cells, and then we want to check how they have actually incorporated the plasmids, the recombinated plasmids that we have genetically engineered. So this is where serodilution may come in the question linked to genetic engineering, or it could come in this case, linked to the fermentation process and biotechnology. So we've been told we're doing serodilutions, and we're doing this to estimate the size of a bacterial population in a culture. In the question, they have told us that we have 20 centimeters cubed, and they have told us that we have 1.0 times 10 to the 6 bacterial cells. Now, this is the new way in which I would say to try and do something when it comes to the fermentation. So if I have a question like this, and I want to look at the population size and what they've told me, I'm now going to do a new thing, which is I'm going to do number of cells. I'm going to put a little line down here, and then I'm going to do the volume, excuse me. Now, I'd like to take credit for this, but actually this was me getting really confused with these numbers. I look at numbers like this and do maths, and I just see it all jumble around. It's just a big mess, and it's just numbers, and it means nothing to me. And sometimes I get so frustrated because I'm like, I'm trying to help students. I need to help them. How can I help them? So I sat down with Alan. I was like, I'm getting stuck. I'm getting this one bit. I can't work out what I'm doing wrong. Please help me. And so he said to me, right, let's try and do it this way. And I didn't like it at all. And now I'm like, oh, actually it works. And so I'm going to show it to you. So he said to draw this table that says the cells and the volume. So I'm going to start with my volume. So I know I've got 20 centimeters cubed, 20 centimeters cubed. And in that 20 centimeters cubed, I have got 1.0 times 10 to the 6 bacterial cells. So I know I've got that. Now, for some of you, writing things in standard form and keeping it in standard form is easy, makes sense to you. For me, I struggle with that. So I sometimes will convert that into the actual number that it represents because it's easier for me to see. It's just because it's how my brain works. Whatever works for you is the way to do it. So what we have is that's the number of cells in 20 centimeters cubed. Now, in the question, they have told me that I have 5% of this 20 centimeters cubed. So if I am going to do 5%, whatever way you want to work that out, I'm going to work out 5% of 20 centimeters cubed. Now, if this was the way I do it, because I do it a long-winded way, I would do that divided by 100 times 5, or I could just have times it by 0.05, whichever way you want to do it. But I've worked out 5%, and that 5% is 1 centimeter cubed. So I have removed 5% of my 20 centimeters cubed. So I have taken 5%. So now what I need to do is I need to do the same thing to the cells. So I now need 5% of that 1 million that I had there. So if I do the same process, that therefore means I'm going to end up with 50,000 cells in 1 centimeter cubed of a volume. So now what I'm going to do is I'm now going to follow the instructions to what it tells me for the next stage. So it now says it's been transferred to a new test tube, and it's been made up to 10 centimeters cubed with water. So I have now added 9 centimeters cubed, and that 9 centimeters cubed means I have a total volume now of 10 centimeters cubed, because I have my 1 centimeter cubed, which had my 50,000, and I've added 9 centimeters cubed. So in a volume of 10 centimeters cubed, I have 50,000 cells. So that's my number of cells in 10 centimeters cubed. So now what I'm going to do is now I'm going to follow the next step. And the next step tells me in the question that I am going to do an additional tenfold dilution. And that tenfold dilution is going to give me a final volume of 10 centimeters cubed. So again, I'm going to take 1 centimeter cubed of my 10. So therefore, if I'm going to take 1 centimeter cubed of my 10, I have, let me change color a second, divided this by 10 to get 1 centimeter cubed. I therefore need to divide this side by 10 to get 1 centimeter cubed. So that therefore means I now have five, oh, I changed my color, hold on. I now have 5,000 cells in my 1 centimeter cubed. And I was told that I'm going to dilute it by adding in my 9 centimeters cubed so that I have a final volume of 10 centimeters cubed. So now what I need to do is the last stage. So the last stage is that they tell me I'm going to take 0.1 centimeters cubed of my final 10. So I now need to get 0.1 centimeters cubed of my original 10. So to do that, I need to divide my 10 by 100 to get to 0.1 centimeters cubed. Therefore, that means I'm going to divide this by 100, which therefore tells me that I have now got 50 colonies, 50 cells, number of colonies, because a colony is represented of a single bacterial cell. I have therefore got 50 in my plated sample. So it doesn't matter what they give you, which way round they give it to you, as long as you understand how to work from one way to another. So if they said instead, I don't have 50 cells, I have, I don't know, let's say I've got 25 cells. So I divided it by 10. So I have a volume up here of 10 centimeters cubed. I need to get 1 centimeter cubed. So 10 divided by 10 gives me 1 centimeter cubed. So if I'm dividing that one by 10, because I'm taking a tenth of it, I need to divide my cell number by 10 too. Did that make sense? I'm going to look for my calculator there. So when you're looking at this, you're looking at, in this case, the sample size. Yes, so a tenfold would mean dividing by, what have you said? Yes. So when it says a tenfold, what I'm saying is I'm taking 1 and I'm adding in 9 to make it to 10. So a tenfold would be dividing it by 10 each time I do it. I'm doing a tenfold serial dilution. So if I went from 1 to another and I take 1 and I've put in 9 centimeters cubed here, whatever was in the original one, so if this is 10, I therefore have 1 of 10. So I have reduced that down to a tenth of the original. If I do it again, 1 centimeter cubed going into the next one, I've reduced it down by another tenth. So now this is 1 in 100. Yeah, does that work? Now it's important that you understand this because what you're going to have to do in the next one brings it in. Now, give me 1 second. I just want to quickly check something. And I will redo this again. So it's important because I want to show you another example in an answer. So give me 1 second. I just want to check. What just happened there? I just moved to the wrong one. Ah, go away, go away. Hitting the wrong button. Okay, so just to go back to this original one, Andreas, I'm just double checking on what you've asked me. Just give me a second. So for those of you who are still looking at this, I was told I had 20 centimeters cubed. I was told that there's 1.0 times 10 to the 6. So that was what I put on this side. I then am told that I take 5% of that original volume. So if I'm taking 5% of this, which is 1 centimeter cubed, I need to therefore have 5% of the cells to work out how many cells I was taking across. So in that case, 50,000 cells is 5% of 1 million. Then I was adding in my 9 centimeters cubed. So now I have 50,000 in 10 centimeters cubed of a volume. I haven't changed the number of cells, I've just changed the volume. So then what I'm going to do is I'm going to again take 1 centimeter cube. So to get 1 centimeter cube from 10, I divide this by 10. Therefore, I need to divide the other side by 10 too. So my 50,000 divided by 10 is now down to 5,000. So I'm adding in another 9 centimeters cubed of water. So my volume, which has 5,000 cells in it, is 10 centimeters cubed. So if I now want to take 0.1 centimeters cubed of that, I'm dividing that by 100. Therefore, I need to divide this one by 100, which gives me my 50 cells. So it's important that when you're doing this, that you note the numbers that they have given you. No, if you are memorizing, that's not going to help you. It's about understanding the content to therefore be able to explain what that means. Because if somebody comes at you with a different question, you need to know how to help them. Yes, Andreas, in answer to your question, yes, that is the case. I wanted to just double check it. Right, okay, so where are we at? No, you can't, nor you can't use them interchangeably. You don't, well, you want to be specific in your wording that you are giving to the exam board to ensure that you are not losing any of your marks. So that's what I would say. Okay, so I want to move on to this next question and explain this next question. So the next one is a student carried out a different serodilution to estimate the size of another bacterial population. So the serodilution results in 4 colonies. So I'm going to again come down to the additional page. So this time, what I'm going to say is if you have 1, 2, 3, 4 samples. Now I'm going to call this A, I'm going to call this B, I'm going to call this C, and I'm going to call this D. Now we're going to say that each of these has got 10 centimeters cubed inside. So each of these has 10 centimeters cubed of a volume. And what we're going to do is we're going to plate 0.1 centimeters cubed of this onto agar. So we are going to plate this out onto agar, and therefore, if we're going to plate it out onto agar, we are going to see how much of it actually grows. So if I plate A out onto 0.1 centimeters cubed of the volume of A out onto a plate, I just get a big splurge. I don't know how many colonies there are because they're just all interlinked. It's too much, it's too big. So what I might do is I will probably need to do a serodilation. So I'm going to do a 10-fold serodilation. So I'm going to put 1 centimeter cubed of A in there. And then I'm going to add in my 9 centimeters cubed of water. So when I do that, what I'm then going to do is I am now going to again plate this to a new plate. Again, I'm going to take 0.1 centimeters cubed. And this time, the growth that I get is I start to see some colonies, but I've still got quite a lot of big splodgy areas that I can't see the colonies distinguishably. So therefore, there's still too many cells for me to use to make an estimate. So what I'm then going to do is I'm again going to dilute this down. So I'm going to take 1 centimeter cubed from B. I'm going to add 9 centimeters cubed of A. Oh, sorry, of 9 centimeters cubed of water, 1 centimeter cubed of B. And again, I am going to plate this out. And this time, I get some clear colonies that I can actually count. So that's how many I have got. And I'm going to do it one more time. So I do it one more time, 1 centimeter cubed. And I'm going to add 9 centimeters cubed. And I am going to remove again 0.1 centimeters cubed. And I'm going to plate that out. And this time, as it said in the question, I get 4. So this is what I've done. I now have 4 in my plate. So if we go based on what we were doing a minute ago, we can go backwards. If I do my cells, my volume, and what I'm going to do is zoom out a little bit so I can write this down. So I am going from the bottom this time. So I have got 4 cells in my 0.1 centimeter cubed. So I want to work my way back up to my 10 centimeters cubed. So to do that, what I'm going to do is to work my way back this time, I'm going to times this by 100. So by timesing that by 100, I now have 10 centimeters cubed of a volume. Therefore, I need to times this by 100. I now have 400 in my specimen, which is in my, why can I not think of the word right now? My test tube. Okay, so now I'm going to go back. This is for A level for OCR A exam board in the UK. However, it is relevant to anything to do with biology if you're doing serodeletions. If you are IB, for example, doing International Baccalaureate, then what I would say to you is this is very good for your coursework. So, Andres, what did you say? If the bacteria tend to form clumps instead of being evenly distributed, how would that affect the number of colonies? So if the bacteria tend to form clumps, then what it means is you can't distinguish them separate enough from each other to class. So what we tend to say is one colony could have come from one bacterial cell. So therefore, we can use the counting of the colony to represent as if we have one bacterial cell. Why do we get more clear colonies after 1,100 dilution? Why can't we see it before that? It depends. It depends on the dilution that you are doing and the original starting concentration. So what I'm doing right now is I'm just showing you what they could give you and how you can work back from it. So it really depends what option they decide to give you in the exam. So I've seen it in the exam where they have done 7 dilutions to get it to the point where you see it. I've seen it where they have done the dilution factor where you are diluting it by only 4 times like this. So it really depends. Now, if you're asking me the question to do with this one, the 0.1 centimeters cubed, that's just because that's what it said in the exam. That question, they could try and throw you off by saying it's 0.01 centimeters cubed that you take. If it's 0.01 that I'm taking, I would therefore need to times by 1,000 rather than 100. So it really depends what they've decided in the exam question that they give you. It's not always that they'll do it, the 100th dilution. So just to be aware, it's whatever's in the question. Use the numbers, use the information. I'm just trying to show you in this case, if we were to go backwards. Yes. So each time we've taken 0.1 centimeter cube out. But above here, we've taken 1. So to go from D, so what I'm saying is, in D, there is 400. Now I'm going to make my way back to C. So if I now want to go back to find out how many, to estimate how many are in C, I would therefore, now I'm not looking at my plate of C yet, okay? There's a reason I'm not looking at it. I'm only looking at my plate D as my evidence. So I need to go back now. So if I want to go back, what am I going to have to do? Well, it was a 10-fold dilution, so I know that I took 1 centimeter cubed and I divided it by 10. So I need to times it by 10 to get back up to having my 10 centimeters cubed here, because I took, remember that 10 centimeters cubed is 1 centimeter cubed of the solution C plus 9 centimeters cubed of water. So therefore, I'm going back. So I'm timesing it by 10. Therefore, I'm timesing my 400 by 10. So I now have 4,000. Why is it 1.1? So I've taken a sample of 0.1 out of my 10. So remember, in here is 10 centimeters cubed. 1 centimeter cubed that had 4,000. So this had 4,000 in 10 centimeters cubed. I've taken 1 centimeter cubed out of that 4,000. So because I've taken 1 centimeter cubed, and we can assume even distribution in the fact that we should have shaken it between each time we've done the dilution, therefore, I now have only put 400 into my D sample. Why are you taking the 0.1 centimeter cubed out? Because I want to plate it. So to plate it, I need to remove a sample, and I would always remove a small sample to do the spreading. So if I'm going to spread, I would aliquot 0.1 centimeters cubed or 0.01 centimeters cubed, depending on which one I want to use. You're welcome. Does that make sense? Has that helped on that front? Good. So just to confirm, so we're now in C and I want to go back to B. So if I go back to B, again, I am doing a 10-fold CR dilution, so I'm going to times it by 10 to get back up here. So therefore, I'm going to times this number again by 10. So I've now got 40,000. So therefore, if I work out how many is in A, yes, that's right, it is for the biotech module 6. Absolutely right. I'm going to go back and times that by 10 again. So therefore, I'm saying A started off, my estimate for A is that it started off with 400,000. Okay, so that is what we're saying when we look at if we've got 4 colonies, it's for OCRA. So OCR is your Oxford and Cambridge exam board. And it's the OCRA. You're very welcome. So I want to do the same thing, but this time I want to do it for C. So if I now do the same thing, I'll do it in a different color. I'll go cells and I'll go volume. If I'm now looking at this and I start at the bottom again, so I start at the bottom, I have got in C, I need to count them 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. 16. So I definitely have 16 colonies in my 0.1 centimeter cubed. So now this is C. So I'm going to go back up to get me into my B. So to go back up to B, no, it's not. That's a lie. I did that wrong. That's in my plate. I want to go back up to C, which is up here. I want to times this by 100. So times it by 100, therefore, will get me to my 10 centimeters cubed, which is in C. Therefore, I want to go up, and that would be my 1600 there. Why isn't 40,000? Is it one? So hold on. So you have diluted it. So we've got 1 times 10 to the minus 1, 1 times 10 to the minus 2, 1 times 10 to the minus 3, then 2. So why have you got, I keep going for my calculator, up there. You're not delusional. You can move upgrades. It's just about how much you put into it with your revision and how focused your revision actually is. Hold on, gotta get my calculator up. Calculator. So solutions will come up in every single year of an exam. It's just how they come up. It depends on, so last year's one was about colonies, but I do think that it could come up as a calibration curve if they wanted to do that, but it really just depends. But it comes up every year. Just how it comes up is the question. So hold on. 1, 2. So what do we do? 1, 2. No, Harry, you don't need to know specific examples for your metabolic inhibitors. So this is, hold on, I am trying to, I'm working it out because you're doing it a different way for me. Sean, I'll answer your question in a second. Let me work on this one, then I'll come back to your question, Amna, on that one as well. So let me just work back. So I want to answer this one question. Working back, we're going to times it by 10 to get back to my 10 centimeters cubed. So therefore I have 1,600. No, I haven't. I've got 16,000 in my B sample. And then if I do the same again, I'm going back up again. I have times it by 10 to get back up to my 10 centimeters cubed. And therefore that therefore means I have 160,000 in A. So the reason I wanted to cover this is because the question here is looking for, and this was 2019's paper, by the way. So this was far enough away that they won't give you exactly the same question, but a similar type question like this, just depends on how they do it. If I am using D, I'm going to end up with an estimate, which is 400,000. Whereas if I'm using C, which has given me the 16 colonies, that therefore gives me an estimate of 160,000 colonies. And therefore, if I make my way back up and look at the actual question, it's asking, explain why the student's estimate of this bacterial population is likely to be inaccurate. If you go with the too small a number of colonies, you are going to have a wider error in the estimate that you've actually collected. So whenever you are going to do your dilutions, you want to look for the one that's giving you clear colonies that are separated that you can count. So we can't count this. We can't count this because it's too big. We can't count this because it's way too big. There's too much in there. So we need to dilute it down. In this case, we've diluted it 2 times so that we can get to the point in which it needs to be for us to get a better estimate of what was in A compared to if we dilute it down by 3 dilutions, 3 tenfold dilutions, that will therefore result in a wrong estimation. So let me just go through what you're trying to ask me, Anna, and get my head around it. So you are saying, so we have gone up by 100, then gone up by 10, then gone up by 10, then gone up by 10. So if we are doing 100 and then we're timesing by each of the others, I keep going to look for my actual phone, choosing my calculator. So no, A is the original. So I haven't diluted A down at all. A is the original, which had 400,000 in it or 160,000 in it. So if we go back up to this one that we were doing up here, our A in a sense is our 1 million. And then we have diluted, no, actually, sorry, A would have been this one, that would have been B, and then we'd added in an extra one, which was C in between. Does that make sense? Has that helped answer the question that you were struggling with? In the maths and biology, just to be clear, the maths and biology is hard. Okay, so because the maths and biology is hard, it means that for some of you, like me, it takes a lot longer for you to understand how to answer the question and how to do the calculation. So you may have to do it again and again and again until you get it in your head. And also finding a way that works for you. Find a way. So for me now, doing it in this table actually makes sense. I can work my way back. And therefore, because I can work my way back, I can figure out how much I should estimate we're in that actual original sample, basically. Now, Sean, to answer your question, how would they do it in a calibration curve? So if you're looking at a calibration curve, you are going to have to do serial dilutions to get known quantities of whatever it is that you're trying to make the calibration curve for. Because if you're trying to figure out how much, let's say, reducing sugar is in orange juice, just throwing something out there, if you're going to do that, you need to have a known quantity of reducing sugars so that you can then look at what they would give you in a colorimeter so that you can then use that to make your calibration curve. And that would then ask you about reading that off. Annie, good question. Has that come up, Joe? It's been a while since I've looked at my spreadsheet because I've been working, doing other stuff. Give me a second. Let me look at the analysis that I've done. No. Did a calibration curve come up last year? Colorimeter experiment came up. Didn't come up there. Zero dilution improvement came up, but no, I don't think it did. Am I going to post my analysis predictions on YouTube? No, I don't believe in doing predictions. The reason I don't believe in doing predictions because actually I change my mind every time I think and see questions and think about papers. Also, it's not easy to predict with A-level papers. And the reason it's not easy to predict is because what they will do is they will just ask a question a different way. So many students came to me. So if I think about my 2023 cohort, they did analysis of the questions. They asked me every year, are you going to do it? I'm like, no, I'm not going to do it. So one student actually did sit down and go through the analysis and they came and they gave the list and said, can I give this to the rest of the class? I was like, sure, if you want to, it's your choice. It's not what I'm going to do. And they had it that this one topic area wouldn't come up. And lo and behold, that topic area came up. Now, students believe going into the exam last year that there wouldn't be that much on the kidneys because the previous year there was loads on the kidneys. But what they do is the way they ask the questions, they ask it in a way which is actually assessing other topic areas, but under the umbrella of, let's say, the kidneys. So they were talking about collecting ducts, for example. So that collecting ducts in the kidneys, but they were actually looking at the hormonal system. And also they were looking at animal response at how the body was detecting the changes to therefore result in affecting the collecting duct. So it's really hard to be able to fully predict something, but what you can do is you can look back and say, right, well, they haven't really tested on that for a while. They haven't really tested on this for a while. So they are probably, for example, going to do something on muscles. I keep saying that. I keep feeling that muscles and neural communication are going to come through quite heavy, but how heavy and how they're going to do it, I'm not too sure. Translocation as well as a potentially more practical based side of things. Maybe I might get it wrong. I might get it right. I don't know. All I can do is go with my gut and look at what I've looked at the research, but they will ask things in such a different way that I don't know on that one. Right. Okay. So I have to get my head around because the way you're writing it, Amna, I hate that. Not because of you, but I hate it because in my head I can't, all I see is lots of zeros like you're showing. So because you're doing it like that, I'm now thinking, right, hold on, how can I make this make sense? Let me, I'm gonna go old school and actually try and do it on a piece of paper for this to work. So if and let me just do this one. So now I'm going. You are very welcome. Right, let me think this through. keep going to look for my calculator and it's not there because I'm using it. So if I do that then, hold on 1 second. I want to double check this. So if I go, go away. So do you need to know gram staining? You need to know and understand the differential staining for OCRA. Gram staining is more other examples like Edexcel and AQA, but it's not, but it doesn't mean they won't give a question about gram staining because they'll link it to the fact of it being differential staining. So they'll give you information about the fact that the gram staining could be for gram positive and gram negative. Right, let me do this again. So if I am doing that right, sorry, I'm gonna give me a second. This is why I'm trying to work this through. So that's when I did it that way. So if I'm doing no, why can I not do this on the calculator? Do you do online or in-person tutoring? Oh, I do it online via Zoom, so it's the same sort of setup as I have on my YouTube channel that I've got right now. Oh, it's going to shut me down on this one. 1 second. There we go. Oh, right. Okay, my brain's not functioning with the question that you're asking. So the first one you have diluted it down by 1, 2. Oh, hold on. I wrote the number of zero. I know you wrote the number of zeros in the bracket. What I'm trying to get is what you're saying. So I know when you're getting confused on it, if we go back to the working back up, which part of it that's confusing you? Because we're timesing it by 100, then timesing it by 10, times it by 10, times it by 10. So you're saying the one in the, so hold on, 100,000 for A. So D, you're saying is, I just went wrong paper. 1 second. A is 1/10 of the original solution. No, the A is 10 centimeters cubed of the original. We're trying to work out how much is in A. No, it is a dilution from A to B, we're diluting it down when we get to B because we've only taken 1 centimeter cubed. Ah, right. Okay. I was literally going, I don't, I'm getting confused here. Yeah. So we're not diluting A, we're trying to find out how much is in A. So therefore, because we're trying to find out how much is in A, that's what we were doing, which is why I wanted to go back to what was in A. So A was our original, which we knew had 10 centimeters cubed. So we were working our way back to find out what was in A and working our way back to find out what was in A. Right, now I feel better now. I feel like I can go to bed and actually sleep tonight now knowing that I haven't confused you more. I was like, hold on, I thought I'd got it, and now you've made me think I've not got it. I'm gonna have to go back and redo this. And I've been literally doing this on zero dilutions for 2 days because I'm like getting myself all tangled up and getting confused. And I'm like, am I doing it right? Am I not doing it right? And I thought, oh yes, I've got a solution. And I was like, to go back to the drawing board. And that's the thing. There are some topic areas which are tough. And there are some topic areas which are easy. And for some people, where maths is an easy subject. So I said to Alan, I'm struggling with this. And he's literally like, oh, it's this, blah, 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 blah. And I'm like, that's not easy. That's hard. You're just saying numbers at me. And that's what happens. So you put up the 1 in the 10th, 1 in 100th. And I was like, oh, there's just too much numbers, too many numbers. All I can just see is like the same number over and over again. I have no idea. And it then freaked me out. And because it freaked me out, I was then like trying to get my brain to activate again. And it's really tough when that happens. But it's good to question and it's good to make sure you know the right answer. And that's one of the things that I would say. So I put a video out today about make sure that when you're finding a tutor, that you do find someone who knows what they're talking about. But we're all going to have some parts of the content where we struggle with it. And so what I do is if I don't understand it, I will then go and do research and I will spend the hours trying to find a way to make it easier for me to be able to do it so that I can then make it easier for students to be able to do it. Now, that doesn't necessarily mean that my way is the right way or that I'll explain it in a way that makes it easier for you. What it means is that I've made it make sense to me. I have made it or tried to make it make sense to the people who are watching it. And there will be some people who don't get it, and that's fine. And there'll be some people who say, oh, she explains it in such a difficult way. That's completely fine as well. But do make sure that you question those tutors, that you're not just taking the information that they've given to you as being gospel. Right. Nana, you've asked me about eyepiece graticules and stage micrometers. So let me just switch a second to a different screen. And then what I'll do is I will bring in a little bit on eyepiece graticule. And stage micrometers for you. So I am on my YouTube channel if you are wanting to actually see what I'm displaying. So this is module 2. Module 2, microscopes. No, it's not that one. It is the previous one I want, isn't it? Is it? Oh, no, it is this one. Okay, so eyepiece graticules then. So if we are looking at eyepiece graticules and we're looking at stage micrometers, then the key thing for you to note is that you are going to need to be able to calibrate the eyepiece graticule. So basically, when you look down the microscope, as you can see here, this is me. Took me a long time to get this photo. It was quite tough. When you look down the microscope, what you're going to do is you're going to see your ruler, and there's 2 different rulers you can quite clearly see here. So one of them is the eyepiece graticule, which is on the eyepiece, which is basically a scale for you to use to be able to work out the size of your specimen when you're looking at it down the microscope. So what you want to do is you want to calibrate that by using this stage micrometer. So I have an eyepiece graticule. I have a stage micrometer. I know that one division on my stage micrometer is 10 micrometers. I know that because it tells me on the side of the slide, on the slide that I'm going to put in on my stage, I'm going to look at my stage micrometer. It will tell me that one division is 10 micrometers. So what I then am going to do is I'm going to line it up. Now I can use any of the lines where it lines up quite nicely. In this case, I've used 4. I could use 2. I can use whatever I want to use. So as long as you have a clear section that you can work out. So in this case, 12 divisions on my eyepiece graticule is equal to 4 divisions on my stage micrometer. So because I've got 12 divisions equals 4 divisions on here, each one of these eyepiece graticules is 4 times 10 is 40. 40 divided by the 12 means each one of these is 3.3 micrometers. So I can then use that to estimate the size of my sample. Now, when you line it up, it's never as easy as that. So here you can see, this is me zoomed into my picture, and we can see I have my stage micrometer, which is 100 small divisions, 1 millimeter. 1 division is equal to 10 micrometers. So I have this stage micrometer, and I can see on my eyepiece graticule that is quite clearly going from number 3 to number 7 there. So therefore, if I have 40 divisions, because there are 40 divisions here in my eyepiece graticule, 40 divisions is equal to 100 divisions on the stage micrometer. So because I know that, what I can then do is say, right, well, with 1 millimeter divided by 40 means that each of my divisions is 0.025 millimeters. Or I could say that I've got 100 divisions divided by 40 divisions, which equals 2.5 times 10 to the times 10 micrometers. Therefore, each division is 25 micrometers. So I can either work it out into millimeters and then convert it to micrometers, or I can work it out into the number of divisions and times it by the 10, which still will give me 25 micrometers. So I now know that each eyepiece graticule unit is 25 micrometers. So now what I need to do is take my sample, whatever that sample is. So here I have a slide, and on that slide you can quite clearly see I have a specimen. And what I'm going to do is I'm going to measure the size of this specimen. So I can see that it is hitting this point of my eyepiece graticule, and I'm going to go to the end, which is this point here. So this is where I am measuring from and to. So I know that I have one eyepiece graticule at 25 micrometers because that's what I measured. I know that this is my distance of the sample size. So my first measurement I'm going to take is that length, which is 64 divisions. So there are 64 divisions in between here in my eyepiece graticule. I then need to times that by my 25 micrometers. Therefore, the size is 1,600 micrometers. Right. Do you know what? It's a funny one because stage micrometers are and eyepiece graticule calculations are something that's very, very specific that's come through more in the last 2-3 years of the exams. So whenever you are doing questions on it, there's not that much for you to use as guidance. So it is important to always go back and just double check what they've asked and practice it. Lots of practice, but actually doing it in the exam, it will be very obvious what they want you to do. If they are assessing it, they're assessing your module 1 knowledge, because it's a practical skill that you should have learned through doing your PAG 1. So I hope that has been helpful, guys. I am going to finish because this is now coming towards my bedtime, and I have been working a very long day today. So, Adam, to answer your question, what's osmosis? Osmosis is the net movement, keyword there, net movement of water from an area of high water potential to an area of lower water potential across a partially permeable membrane. So it's important that you remember that it's to do with the water potential from a high water potential to a lower water potential and across that partially permeable membrane, because you've got some channel proteins that allow the water through. Why do you need that? Well, you know, because the phospholipid bilayer has the hydrophobic region, it will repel the water, and therefore it is only partially permeable because of the channels. Guys, have a good evening. Thank you for joining me. I will be back, if not on Monday at 8:30. It might be a bit later, it might be 9 o'clock. But yes, I will be back on Monday night. So thanks for joining me.